Hi everyone, today I want to discuss the different recombinase systems used in mouse genetics. We will cover tissue-specific and inducible recombinases, and for each one I will explain their primary use. Before that, let's briefly recap what the recombinase systems are. Today we will talk mainly about the LoxP recombinase system, which is the most used in mouse genetics. The Cre recombinase is a protein naturally occurring in bacteria that recognizes specific sequences known as LoxP sites and swaps the DNA between them. So how does the Cre-LoxP system work? So you have one DNA and you have this LoxP site in the middle, and let's call this section of the DNA A and B. And then you have another piece of DNA that also has a LoxP site, and let's call whatever is upstream the LoxP site C and whatever is downstream D. So if we add here in the mix a Cre recombinase, what will happen is that this A segment will be followed by the D segment, and conversely here the C segment will be followed by the B segment. And of course we have the LoxP sites in the middle. So far so good, but how are the Cre-LoxP systems used? So the main use is the following. Imagine that we have here the genome, and we have some section of the genome that we call section A that is followed by the Cre site, and then we have a situation where we have a segment that we call B that will come this way and then will follow this way, and after that we have a section that we will call C, preceded by a LoxP site. So we have A, we have B, and we have C. So if we repeat the same that we have here and we add a Cre, what we will end up is in a situation where we have the genome coming with this section A, the LoxP site. As in the previous example, this will be followed by section C. We will go from here to here, have section C, and here is the continuation of the genome. And what will happen is whatever is in the middle is like removed, and it's creating a circle that corresponds to section B. So eventually this will be lost when the cell divides, and we will have effectively a deletion here of this section B in the genome. So this system can be used then to delete specifically one region of the genome. So that's the main use that is given to the LoxP Cre recombinase system, but there are some variation, and I think it's worth mentioning them because it helps also understand how the system works and how you can adapt it to other circumstances. So let's see some other uses of the system. The first one is flipping. So for example, let's say that we have here a gene with a promoter, and we put here a LoxP site, and then we come and we have a gene that, let's call it GFP, and after that we have the red FP, but not in the same strand but in the opposite strand. So this is the RFP, and then we have a LoxP site going in the other orientation. So here I want to highlight the importance of the orientation of the LoxP site, because previously when we have it in the same orientation, this section gets deleted. When we have facing each other, what happens is the following. So when we add the Cre to the mix, what we get is the very similar situation. But what happens here is that the genome, the DNA will come from this side, and then it will continue going in this side, and then finally it will incorporate this last bit. Effectively what you end is with the RFP first, and then the GFP in the opposite strand, so GFP. So this system allows you to have the gene expressing the GFP when you don't have the Cre, and once you have the Cre you can mark the cells with red FP because the sequence will be flipped, and in this case the red FP will be produced. So this is the flipping usage that you can give to the Cre system. The other aspect is the differential use of LoxP sites. So there are different variations to the LoxP site, and you can take advantage to make different clones within your population and have a chimera of cells. So let me show how this works. So this is the selective deletion strategy. And in this case, for example, a classic way of using this is, again, you have your genome, you have a promoter, then you have a classic LoxP site, then you have a variation that we will differentiate it. This one would be fully marked. And then you have one gene, GFP, then you have here the end of the gene with the poly A signal. Here we have the variation, the black variation of the LoxP site. Then you have the red FP, this time in the same orientation with the end of the gene. And then you have the other LoxP site, empty LoxP site, followed by the yellow fluorescent protein, and again the end of the gene. So without Cre, this loci will be expressed in the GFP. But if we add the Cre, two things can happen. This LoxP set can be used, or the empty one can be used. So you have these two alternatives that will lead to two different outcomes. So in the first outcome, we will have the deletion of the GFP, and then we have the red FP and the yellow FP. So we'll have that side there. And actually, in this system, these LoxP sites are inverted. Sorry about that. So the empty one is internal, and what you have is the removal of all of the section, and you have the RFP, the end of the gene, and then you still have this other empty LoxP site, the YFP, and the end of the gene. And in case where we have the deletion of this LoxP site, the other one, what we are left only is with the YFP gene. So we have the promoter, we have this empty LoxP site, we have the YFP, and we have the end of the gene there. So if we don't have the Cre, we are expressing the GFP. In case we delete with these two LoxP sites, we will have the RFP, and in case we delete with these two here, we have the YFP expression. And note here that importantly, there is these different types of LoxP are incompatible, so we either go this way or this way, and there are no other combinations in this case. So these strategies are used for identifying individual cells in different lineages, as I mentioned. If you want to study this in more detail, you can check the Mice Rainbow paper that published in Nature Methods, and if you go to PubMed, you can access this link for the full text if you are doing this for academic purposes. In addition to the Cre-LoxP system, there is another commonly used, that is the FLP recombinase and FRT sites. So this is also used, and again, the FLP sites are incompatible with those LoxP sites, so you cannot use this Cre to delete LoxP sites. Now let's dive into the different types of Cres. The most common type of Cre is the tissue-specific one. In this case, we are not changing the Cre itself, but rather we are restricting where it is expressed in the mouse. The main idea behind the Cre recombinases is that we cannot only use it to remove a gene, but we can remove a gene in some cells while the rest of the animal remains intact. In this way, we can dissect tissue-specific effects from systemic effects. So how do we do tissue-specific recombinases? What we do is express the Cre under the promoter of a gene expressed specifically in the tissue we want to induce the deletion. So there are different types of tissue-specific Cres. The traditional one and most common probably now is the Transgenic Cres. So in this case, we have a construct where we have our Cre and we have the promoter of gene X that will be driving the expression of this Cre. So then we electroporate this construct into the cells, and this will integrate somewhere, somehow. We don't know exactly how this typically occurs, and we will end up with some unknown part of the genome that has this promoter from our gene X, followed by the Cre coding sequence. These alleles work incredibly well, but since only a section of the promoter is used, the expression of the transgene sometimes differs from that of the original gene. The other aspect is that we don't know where most of these Cres are inserted in the genome and how these may affect the expression of other genes. Because of this, it is typically recommended to keep the allele in heterozygosity. In other words, we always try to avoid breedings where both the male and the female have the allele. One way to control for one, not disrupting other genes, and two, having better control over the expression of the Cre is to insert the Cre in the locus of the gene that we are using to determine the expression. In other words, instead of taking the gene's promoter and adding the Cre is directly targeted into the gene with the tissue-specific expression. And these are the Cre knock-ins. So let's see how they work. So we have the knock-in Cres. In this case, we were working on the gene X loci, and we will have a typical genome structure, promoter, followed by exon 1 that has probably ATG there, then exon 2, exon 3, and the rest of the gene. In this case, we generate a targeting construct with homology to the first exon, in particular this part before the ATG. And then after that, we insert the Cre and we add a poly-A signal to stop transcription so that there is no more transcription of the original gene. We can add some homology here to facilitate the insertion, but no more. And so this will be electroporated. There will be recombination here, and we will end up with this allele where we have in the locus of gene X, the promoter of gene X, and all other regulatory elements. Then the first non-coding part of the gene X, ATG, followed by the Cre recombinase, the poly-A signal, and then the rest of the genome here, given by exon 2, 3. That will not be expressed because we are going to put a polyannulation signal over there. In addition to the tissue-specific Cres, we can also control the temporal expression of the Cres. The most common way to achieve this is to use the ERT2 Cre fusion protein. Let me show you how it works. So let's talk about temporal Cres. So we'll have our Cre inserted in a specific region of the genome. Typically, this Cre is inserted in the ROSA26 loci. This gene encodes for a non-coding RNA that is expressed in almost every cell of the body, and therefore you can have your Cre expressed in every tissue that you want to study. ROSA26 doesn't seem to be playing any physiological role, so you want to keep it also in heterozygosity, but it's not a critical gene. So you have the Cre then inserted after the ROSA26 promoter, but this will be a fusion protein where we have first the ERT2 part and then the Cre itself. The ERT2 comes from the estrogen receptor gene, and this is a part of the estrogen receptor that binds estrogen, and when it's bound to estrogen, this protein changes in such a way that it translocates from the cytoplasm to the nucleus. Instead of having specificity for estrogen, it was mutated in such a way that instead of binding estrogen, it binds another molecule called Tamoxifen. And the two come from the second iteration of these mutants. So you have your gene, then this gene produces an RNA or mRNA to be more precise. So this will be the mRNA. The mRNA then will be produce a fusion protein. So this will be the ERT2 side, and then you have the Cre fused to it. And so this part of the process will be occurring in the cytoplasm of the cell. Here I'm making the whole cell, and here we have the nucleus, and this is the cytoplasm. So what happened is when we add tamoxifen into the system, and this you do by injecting the mice with tamoxifen, and here you can define exactly when you're going to do, and we are going to represent tamoxifen with this square. So you can add this into the cell, then tamoxifen will come here, will bind to this complex that is in the cytoplasm. One is bound to tamoxifen, the complex translocate to the nucleus, and in the nucleus it will find a LoxP site. So for example, we can say, just to keep consistency with the previous example, we have 3 sections flanked by LoxP sites. Let's call them sections A, B, C as before. With the Cre in the nucleus, then these sections becomes A followed by C. So we are deleting the part B, and then B is eliminated from the genome. And this is how you can eliminate whenever you want a gene from the genome using this tamoxifen compound. As you can imagine, you can do this not only with the ROSA26 locus, but you can have both the temporal and the tissue specific Cre, where you insert this construct in whatever gene you are interested here. Let's call it gene X. This technology is almost 35 years old, but is still probably one of the most robust. This is the original paper from PNAs that explains it in details. Okay, the last thing I want to cover today is how to generate null animals. The null animals allow us to do an unbiased assessment of what physiological processes are most dependent on the protein we are studying. The idea here is to generate completely null animals. To make sure that all the cells in the bodies are converted, we first generate animals that transmit the null alleles to their progeny. Let me explain what I mean with that. So we are covering how we are generating null animals. So we want to have a breeding here where we have our floxed allele in heterozygosity with a wild type allele, and this animal will not have any Cre, and we will cross it with another animal that will be wild type for the loci of our Cre, but will have a Cre recombinase in such a way that we have a progeny, and this progeny will be a chimera. So some cells will be plus flox, Cre positive. There are other combinations, but in case that we inherit the flox and the Cre, and in some other cellular cells we have the recombination and also Cre positive. So these animals are effective chimeras, and the idea is that this conversion occurs in the germline. So in the cells where this Cre is expressed, we will have the conversion. In those that is not expressed, we won't have the conversion, the flox allele. And then we will cross these animals with ones that are completely wild type, both for the loci of interest and the Cre. And then, so we will have an animal that, if the germline has been converted, can transmit the null allele, and then we want to get rid of the Cre. So we have an animal that is heterozygous for the mutation, and then we can cross this animal to another one that was generated in a similar way. So which are the different strategies to generate germline depleted animals? First, you can try depleting in most of the tissues, including the germline, or you can target specifically the germline. So just to give some examples, for example, we have like the morula, one of the first stages in embryo development. Then you have, the blastocyst there, it's a mouse blastocyst, and then we have a mouse with the germline over there. If you use a Cre that is expressed in almost every tissue, such as the CMV Cre, you have deletion from very early on. So all of the tissues of the animal, including more importantly the germline, have the deletion, and all of those animals will be transmitting the allele. The other strategy is to do a germline specific. So in mammals you have the specification of the germline, not in the morula or zygote, but it's a little bit later in development. So you can target directly these cell lines. Whatever you want to target, the important thing is at least the germ cells are targeted. This is a mouse. And you can either target like the whole germline or the male or the female germline specifically. So for example, DDX for Cre targets both the male and the female germlines. Then you have like the straight that targets only the male, and you have others like the ZP3 Cre that targets the female. Anyway, any of these ones can do the job. So on a note on the different types of alleles, the best websites to get information from floxed alleles, one is the mouse genomic informatics. Here you can put any allele. I put one of those that we work on. Gives you the gene, and then you can click the alleles, and it gives you all the alleles that were generated for this particular gene. The other good website is the JAX laboratory website, where it gives you the information for each of these strains. So here we have the CMV Cre, and it states CMV Cre mice are useful for the depletion of LoxP flank genes in all tissues. The transgene appears to be X-linked since transgene transmission through males is restricted to female offspring. So this is one of those genes that was generated as a transgene. So we don't know where it is exactly. And it's used in these virus promoters so that every cell is exposed. So these two sites are great for both finding LoxP sites and also finding information about the Cres. Okay, that's it for today. Now you know how the Cre-Lox systems work. And we learned about tissue-specific Cres, inducible Cres, and how to generate null alleles from floxed alleles. If you find the video valuable, please subscribe to the channel. It's the best way to make the content on the channel more accessible to people like you, including yourself in the future. If you are interested in other aspects of mouse colony management, check the playlist now on screen, and I will see you there.